Bioinfocongress IV. Dr. Ming Tang. Learn computational biology the right way. Dr. Ming Tang is now the Director of Computational Biology at Immunitas Therapeutics. At Immunitas, they employ a single-cell sequencing platform to dissect the biology of immune cells in human tumors. Dr. Tang is a computational biologist working on genomics, epigenomics, and single-cell transcriptomics. Together, Dr. Tang has over 10 years computational biology experience and 6 years wet lab experience. He uses R primarily for data wrangling and visualization in a tidyverse ecosystem. Dr. Tang uses Python for writing snake-make workflows and reformatting data. He is a Unix geek learning shell tricks almost every month. He cares about reproducible research and open science.
Dear Dr. Ming Tang and our participants, welcome all of you to our Bioinfocongress IV events. Dr. Ming will give us a talk today on Learn Computational Biology the Right Way. At the end of the presentation, we will have a question-answer section prepared by our team and consisting of questions from our participants. Dr. Tang, welcome back.
Thank you very much for the invitation and the introduction.
You're welcome. If you wish, you can start your presentation.
Sure, I will do full screen share slideshow. Can you see it? Okay.
No, right now. So we can see it.
Okay.
If you wish, you can start.
How about now?
Yes, we can see it.
Okay, so I will go ahead. First of all, thanks for the invitation. It's my honor to present at the Bioinfocongress 2023. My name is Ming Tang. You can call me Tommy. So I'm currently the Director of Computational Biology at Immunitas, a single-cell genomics company committed to develop novel therapeutics for cancer patients. So today I'm going to talk about learn computational biology the right way. I think my talk is right at the right timing because our last speaker, Dr. Ahmed, also said learn how to code as soon as possible. So I hope this talk will give you some inspiration so you can learn how to coding as early as you can. And I hope my experience can, so I'm going to talk about how I learned it the hard way, but I want to show you the right way. And I hope my experience can help you to avoid the mistakes that I made. And this is my Twitter. I tweet mostly about bioinformatics, the tools, the papers, and some tips. Sometimes I also complain about it, about bioinformatics. And this is my blog, and I post some of my learnings there too. So in this slide is a schematic view of my career trajectory. So I graduated from Shanghai Jiao Tong University in 2008. And I started my PhD at the University of Florida. And then I was trained in a wet lab. So everything was fine. It was great. I had my 2 first author publications in well-respected journal at 4th years of my PhD, not until one day when my advisor asked me to analyze a public ChIP-seq data back in 2012. I couldn't open it with Excel, so I was forced to teach myself Linux, R, and Python. So I then did a 3-year computational biology postdoc with Ro Vahak, who was leading the GCGA glioblastoma project. And after my postdoc, I moved to Harvard FAS Informatics to work on single-cell RNA and single-cell ATAC sequencing data. So in 2020, I joined Dana-Farber to lead an NIH-funded consortium project called Cancer Immunologic Data Commons, in which 4 cancer centers carry out immunotherapy clinical trials, and they profiled patients using whole exome sequencing, TCR sequencing, RNA sequencing, and ATAC sequencing. So our job was actually to process all this data securely over the cloud and give them back for downstream analysis. So I stayed in Dana-Farber for almost 2 years, and now is leading the computational biology team at Immunitas. So I have a great interest in promoting open science, transform biological data science education by teaching. So I consider myself as a genomic data scientist or a computational biologist. So I have extensive experience in analyzing sequencing data for genomics, epigenomics, and transcriptomics. So I use R primarily for data wrangling and visualization in the tidyverse ecosystem. I use Python for writing SNCMake workflows, reformatting data, and Laravel for the Unix commands. I share my experiences of sequencing data analysis in my GitHub repository. So you can go there and see, find those resources. It will be helpful for you. And the reproducible research topic is always near and dear to my heart, and I will touch on this topic later. So I just want to show you the transformation using these two pictures. So in 2013, I was pipetting on the bench, doing qPCR, cell culture, and Western blot. And in 2023, I am a computational biologist. So if I can do it, you can do it too. So it takes me almost 10 years, but you can do it shorter with a clear roadmap. So we are in the age of data exploding, right? This is an old stats from Sean Davis. So this is how much basis we have back in 2018, 1.8 times 10 to the 16 basis in the NCBI sequence read archive. This is equivalent to 6 million human genomes, which is 9 billion bases, 3 billion bases. And every day, they process roughly 3 TB data. And this is really old, like 5 years ago. And nowadays, you can imagine the data is exploding even more quickly. And now we have single cell data, image data, spatial transcriptome data, and it can be overwhelming without any data skills. So I believe many of you work with data, but which category are you in? So I really like this cartoon borrowed from Thorson, who is a bioinformatics professor in Australia. So if you know computer, and stats, you are a data scientist. But if you happen to have the domain knowledge, in this case, biology, you are a Superman or Wonder Woman. I would argue actually a bioinformatician or computational biologist should know all the 3 fields. So I will use bioinformatician and a computational biologist interchangeably. There are some differences, but I'm not going into the nuances. So what should you learn to tame the data? So my first tip here is you need to learn Unix command line. So humans and computers commonly interact in many different ways, such as through a keyboard, a mouse, a touch screen, or using speech recognition systems. And the most widely used way to interact with personal computer is called a graphic user interface, or GUI, if you happen to heard that before, GUI. So with a GUI, we can give instructions by clicking a mouse and using manual-driven interactions. The Unix command interface, or the CLI, is a scripting language allowing such repetitive tasks to be done automatically and fast. So really, why we need to use command line? So remember, the text form, a file, is still the key format of bioinformatics. And most of your work will be actually wrangling the file formats in order to feed into different bioinformatics tools. And Unix commands are perfect for wrangling the files. And most bioinformatics tools are run by command line. So if you don't know how to use command line, you can't use them, right? And it's more efficient and powerful with a single command, like here, copy all the PNG files into this picture folder, for example, this simple example. Instead of clicking all the PNG files and select them all and drag them into this picture folder. This is a simple example. And the Unix command line is omnipresent in many settings. For example, the high performance computing cluster, and cloud computing, they don't have the GUI interface. So you have to learn how to use Unix command line. So this is how it looks like a terminal. So essentially, a terminal is a command line interface, and this is the GUI interface that we are familiar with all the folders in here in my home directory. And this terminal, it's like a black box, right? You type in the commands, you press enter, and it's executing that command. But you will see the same file system represented in the terminal. So all the folders here will be the same as here in your files in your GUI file system. So I recommend either using a Mac or you can have a dual booting system for Ubuntu to have this unique command line interface. And this software carpentry has a lot of nice beginner courses to teach you how to use this Unix command line. The next tip is to learn some Python. Python is the most popular language in data science. It is easy to learn and is really powerful, right? So why in this cartoon it says, well, you are flying how? This guy said, oh, I'm using Python, right? And the good thing about all those languages, there's so many libraries or packages that you can use. So in this case, you just type import anti-gravity, and then this guy is flying. So take advantage of those nicely made libraries. So next is to learn some R. So R is a statistical programming language, which is free, open source, and powerful. And R has its strength in statistics and plotting. So for IDE, I really like RStudio. And for R packages, don't try to reinvent the wheels. So use the tidyverse and ggplot packages for data wrangling and do this and data visualization. So Bioconductor essentially contains many different R packages specifically for bioinformatics. So if you search an assay name, then add a bioconductor, you mostly likely you will find a package that is written by someone else. So this is the data analysis workflow from this R for Data Science book. So first you import the data, you tidy the data, clean the data, then you explore the data, then you communicate. I want to emphasize tidying the data is one of the key skill sets actually in data science. I would say 80% of the time I spend is to clean up the messy data. So again, this book is also free to read, so go there. And investing your time to learn a tidyverse is a good investment. Data visualization is very important in exploratory data analysis. So you get an idea of how the data look like. You can review the structure of the data, identify if there are any batch effects or sample swaps in your experiments. For example, in this example, like all those points, they all have the same mean, the same standard deviation, and also the same correlation. But if you visualize data, they look drastically different. So learn how to use ggplot2 or this R package, and then you can do a better job in exploratory data analysis. What people think we do usually, so they think we have a magic push button bioinformatics keyboard. So I borrowed it from Thorsten again. And we have just, assemble the genome, I just press this key, and covariance, I just press key. But in reality, what we do, so this is a typical day of my life as a computational biologist. The first thing is to install software. Second is Google how-tos and the error messages. Then read the manuals of bioinformatics tools, converting file formats, as I mentioned before, tidying the data 80% of your time, The real analysis is like only 20%. So really, Google is how we learn and do things. So in this example, it's very funny actually, cartoon here. So Rob, you use Unix, there's a bomb that's going to explode. To disarm this bomb, you simply type a valid tar command and in your first try, and no Google around. But then this guy failed. I'm so sorry. So this really shows that we use Google a lot. Even at some commonly used command, we probably cannot remember the exact syntax. So it's okay to Google. And it's even better to know some of the Google tricks. So I will share some of those with you. For example, if in Google, if you add these brackets and the small letter R, it will search all R programming related pages. And if you use like bracket and use Python, and it will actually find you the Python related web pages. So you can use this double quote to search for exact phrase. Use this tilde sign in front of a word to find synonyms. And if you can use this minus sign to exclude the terms that you don't want to search. And you can also even search within a specific website. For example, you want to search heat map. You want to know how to make a heat map, but only within the site of this bioconductor supporting page. So this will search a heat map inside this website alone. You can also define a file type that you want to search. For example, heat map, then you say file type colon PDF. It will only give you PDF files in the searching results. So I hope those little tricks can help you. And again, so ask for help. So no one knows everything. And those are like 4 really popular forums to find help. Seek Answer, BioStars, Stack Overflow. Those 2 are more like sequencing data related. Stack Overflow is general programming language, and Bioconductor helps support, the help page is for bioconductor related questions. And by the way, so when you ask a question, make an effort to ask good questions. So you have to define problem, what you have tried, what did not work, and it's even better to include code. So many times I see people asking questions online like, this didn't work, and that's it. So if you don't give enough context, people cannot help you. So again, if you do not make an effort to ask good questions, don't expect other people to make an effort to answer you. That's fair, right? And I'm not lying, right? And this professor, I think is from Stanford, he was trying to find out something, Google something about snakemake. And actually, he found an old post that he actually was asking the question. And then he got the answer again, for sure. So now I will switch the gear a little bit to talk about Excel. So we really want to use Excel with precaution. I know Excel is still the most primary tool that people use for data analysis, but we need to be aware of its weakness. For example, in this news, it says some of the data are lost, COVID-19 data are lost because the Excel, this old version of Excel can only like contain like 1,400, for example, rows. And in this example, actually, an Excel screw up leads to a retraction. Actually, it causes the rows to shift the data from different groups. And that's actually tell you something, because if you use Excel manually, it's really easy to make those type of mistakes. What's even worse is that Excel converts gene names to dates. And some examples here, for example, APR1 will be converted to April 1, OCT4, OCT4, which is a really well-known transcription factor, right? But it got converted into October 4th. So really be aware of this problem. And it's prevailing in all those published papers in their supplementary file. So you really need to learn, for example, how to use R packages, read XL to read in the Excel file, and use R packages such as Janitor to actually clean the Excel file. So now let's dive into my favorite topic, reproducible research. So we all know how science should be done. However, you first make an observation, right? And then you form a hypothesis, perform the experiment, analyze data, and report your findings. And then you invite others to reproduce the results. But however, we often hear that one cannot replicate a published study. So there's a reproducibility crisis going on here. So according to Titus Brown, most computational research is not reproducible. So you may think experimental studies may be hard to reproduce because the experiments were performed under different environments, different people handled the mice, or different reagents were used. You may think computational studies should be easier to reproduce. However, according to Titus, a bioinformatics professor at UC Davis, 95% of the studies fail to include details necessary for replication. So it's really hard to build off research like this. So let me give you an example. So there was a scandal in Science that the researcher at Duke faked the data and built the results on the fault analysis. And Dr. Kate's group at MD Anderson and downloaded their microarray data and could not reproduce the results claimed in the paper. So through a detective investigation, he finally found original authors had one row shifted in Excel and got all the gene names wrong. Unfortunately, the clinical trial actually built upon the results has to be stopped. This speaks to the fact that how important we should make our analysis reproducible, both for your own sake and for the patient's sake. So I highly recommend actually you watch this video, and it's very fun. I will actually share this slides to the organizer, and then you can get the slide deck. So here's another example. In this paper published in Science, the rearrangement bursts generate canonical gene fusion in bone and soft tissue tumors. And in their method section, they say we detect gene fusion in regions of genomic complexity using an approach that integrates multiple independent fusion algorithms. And that's it. So without knowing the details, I don't think I can reproduce their results. Again, method matters. So why reproducibility is so hard? So there's several reasons that I come up with. But you think about on your side. The first is no raw data available. So they didn't give you the raw data. And second, scripts and data available upon reasonable request. And then you still see this actually in the published papers, which in my opinion is unacceptable. Third is a lack of method description, just like the Science paper. The version of the tools may be different. So because if you use R or Python, different bioinformatics tools, they may be built in different versions. And they can generate different results. And lastly, you may run in a different machine. The Unix machine versus a Windows machine may give you some different results. So if reproducibility is so hard, should you care about it, right? But keep this in mind. You are going to do the same analysis for sure in the future yourself, especially during the journey of submitting a paper, right? You're going to get the comments from the reviewers, and you're going to actually redo some of the analysis. And this is really for your own benefit. And now I'm in a biotech company. Our mission is to develop life-saving drugs for patients. I certainly want my analysis to be reproducible. Then how to ensure reproducibility? There are several points I wrote it down here. So first, Git version control. Second, using a notebook, Jupyter or R notebook, document every step. And you can use containers, like Docker containers, Singularity containers, which is used to containerize your computing environment so others can reproduce your analysis using the same environment. If you are doing software engineering, you want to do unit test and continuous integration of development, CI/CD. So I will talk about some of those points in detail in the coming slides. So I'm sure we all experience this on versioning our Word documents. We just keep adding version numbers after final, like final revision 2, 3, 6, and then it becomes longer and longer. And Git is a software for tracking changes in any sets of files, usually used for coordinating work among programmers. So Git records all the history of your file, so you can easily revert back to any previous version. And GitHub and GitLab are websites that host your Git repositories. I highly recommend putting your code for the published paper on GitHub so others can replicate your analysis. And there's some learning resources here in this link. And let me talk about notebooks. So a wet lab member has a notebook to document his daily experiments. So what, when, and how he or she performed experiments, right? Similarly, a computational biologist should have a digital notebook to document all analysis. And Jupyter Notebook is a Python-flavored notebook, although it supports other kernels such as R. The nice thing about notebooks is that one can put the documentation together with the code. And the code is evaluated in line. So I usually have a notebook for every analysis that I did. So how I download the data, where I download the data, what command did I use? So all put them in the same notebook. So I use actually R more often. So I love R Markdown or R Notebook. It's similar with Jupyter Notebook, but it's more like R focused. Again, you can evaluate those commands in line. For example, there's a figure that is generated included in this notebook. So here I want to talk about automation. So if you want to make your analysis more reproducible, you want to automate your analysis as much as possible. Not only it helps you to make your analysis more reproducible, it also saves you time in the long run. So in this figure, I'm showing you there are two ways, the geek way and non-geek way to approach certain tasks. So when the task size increases, the geek way is that, oh, I get annoyed. Let me stop and write some scripts to automate it. So it takes some time. But then after he has it, he can run the scripts. And when the task size increases, and it actually is better than the non-geek way, which does all the tasks manually. And remember, computers are really good at repetitive work. And the good side effect of automation is you don't need to actually document it. The best documentation is automation. So write scripts for everything, unless it's not possible. If you have to manual editing, document it in a notebook. Say, oh, I did this, did that. Document, document, document. So important things I say 3 times. And we also use this, write all this document in Markdown files. And this tool called mkdocs, which translate all your Markdown files into a really nice website. And there's some tips for automation. For example, if you have a repetitive simple task, put them into a shell script, for example, myroutine.sh. So instead of typing each individual command every time, you can just run myroutine.sh. It can save your time. And you can also use the old GNU-make. Once was designed for compiling software, but people use it for data analysis too. Some more recent workflow language such as SnakeMake, Nextflow, and Widow are your friends too. And in this repo, it collects hundreds of tools that can be used to write pipelines. I would suggest you just start with one of them. I highly recommend SnakeMake, and I know Nextflow is drawing attentions to. SnakeMake is just a Python extension, so if you know Python, it will be very easy for you. So let's talk about containers. So why we need containers, right? So imagine you are working on an analysis in R, and you send your code to a friend. Your friend runs exactly the same code, exactly the same data set, but it gets slightly different results. So this can have various reasons, such as different operating system, a different version of an R package. Docker is trying to solve problems like that. So you might have you use the VirtualBox. Think it as a virtual machine that encapsulates all the software, libraries, and configuration files. Then you can ship this virtual computer to your friend, and you can use the same computing environment to do the same analysis. And this in part solved the reproducibility issue. And I also put two links there for you to learn. So as I mentioned, the number one actual problem of bioinformatics is converting formats of input and output between different tools. But the number zero problem is installing tools. So there's so many bioinformatics tools, and many of them have different dependencies on other software. For example, a particular tool is Python 2, and another tool is Python 3. How can you do the analysis if the analysis is dependent on both tools? So Conda actually is a Python, is a package management tool for any language. And you can set up two different Conda environments, for example, one for Python 2, the other one for Python 3. And you can activate different environments for different tools. And Bioconda is a life science-centered channel in Conda. And so my message here is to use Conda to manage your computing environment to help with the reproducibility. So project organization is really underrated, and not many schools teach how to organize a computational biology project. So I recommend you to read this paper, A Quick Guide to Organizing Computational Biology Projects. And I follow a similar folder structure. So on the top level is the name of the project. Then you have a documentation folder, a data folder, a source file folder, the bin folder, and the results folder. And you prefix all the results and the data folders by date. So you don't need to actually follow exactly the same folder structure, but as long as you have a consistent folder structure, that's fine. Having a fixed project structure can help you to reproduce your results. There are two more papers that I recommend you to read. It's Good Enough Practices in Scientific Computing and the Best Practice for Scientific Computing. So they were written by the software carpentry community. I highly recommend you to read them. So one can take even one step further. So this is a tool called RPack called Workflow R. So it's an RPack to translate your analysis R Markdown files to a web page. And one can easily browse through the code, the documentation, and the figures on a website. And you can share this website to everybody. It's very, very neat. This is an old post that I wrote back in 2015. Some tips for coding from a bioinformatics beginner's point of view. So first, computers make mistakes. They can give you nonsense results and exit without error. So you have to actually make extensive tests before running your code. Also, do sanity check with the results, right? Check with whether the result makes any biological sense. Talk to the experts in the biology field too, whether this makes sense or not. Second, share your code, right? Even your code is correct, you want to share them so other people can look at them, and they also may improve the code. So you want to make your code reusable. So you do not want to hard code your scripts. If it takes a file path as input, make it as an argument in your script. So you can use the same script for different analysis. You also want to modularize your scripts. So usually the data could come, for example, come in different formats. Take chip sequencing as an example. You may have a script that starts processing the data from FASTQ to the final peaks. And you want to modularize the scripts into two modules, for example. One for mapping the FASTQs to band, and the other is from the band to peaks. So modularized scripts can help you because if the data come in the band format, you can directly use in your second module to call them peaks. 5, heavily comment your scripts. So it will not only make other people to understand your code better, but also make the future you to understand what you did. To trust me. So your future self will thank you if you comment your scripts. And lastly, you need to make your analysis reproducible. And how to make them reproducible, I have actually mentioned in previous slides. I think this is my second last slide. There's some key takeaways from this presentation. So if you want to learn computational biology, first, you need to learn Unix commands, Python, and R. Second, learn how to use Google effectively. And even recently, we have this ChatGPT. I'm not sure whether you're aware of this. It's an artificial intelligence language model that can understand and generate human-like language, making it useful for tasks such as conversational AI, language translation, and text generation. And you can take advantage of this to learn computational biology. You can ask him questions. Third, be cautious with Excel. So you still use it, but remember the weakness of Excel. It may actually convert your gene names. It may actually also truncate your data, for example. Git version control your code. So you can always track how your code is evolving, and you can quickly revert back to your old code if the new code didn't work anymore. You need to have a consistent folder structure for projects, reproducible computing. And learn by doing, I didn't actually talk much about it, but the way I learn computational biology is really learn on the job, which means if you're in a wet lab, but the lab is doing some sequencing experiment, take that project and then learn how to analyze this sequencing data by yourself. And I learned how to analyze chip sequencing data because I had to analyze the public available chip sequencing data. So learn by doing. And lastly, I want you to focus on your strength. I know we are not a computer scientist, we are not a statistician, but really biology, the domain knowledge is our strength. So I will focus on your strength, but gradually learn other skill sets, for example, the computer languages, programming languages, and the stats. And this is my last slide. I want to thank Samir Emming, my colleague during my postdoc in the Hack Lab. He taught me a lot of new things when I was a beginner. Titus Braun, a professor in UC Davis. I attended his next generation sequencing workshop back in 2015. And Data Carpentry actually has a lot of educational free courses for you to learn data skills. And I also want to thank all the people who share their wisdom on the web. Thank you very much. And that concludes my presentation. So what questions do you have? I will.
First of all, thank you very much for your very informative speech. I have questions from our teams and from YouTube participants.
Okay.
And my first question is, how can we measure whether we have enough data sets for an ideal analysis where we can use computational biology mechanisms?
How to measure the size of the data sets?
How can we measure whether we have enough data sets for an ideal analysis where we can use computational biology mechanisms?
Okay, I think, thanks for the question. I think it really depends on which field exactly you are in. For example, like if you are in for sequencing data, and I would suggest you at least have, for example, for any sequencing experiment, you want to at least have duplicates. Better to have triplicates. I mean, the more the better. But I mean, of course, it's expensive to generate those data, but you need to have like enough samples to have statistical power. And you can always calculate the power of your experiments beforehand. So always talk to a bioinformatician or statistician before you carry out any experiments. So in order for your data to be usable.
Thank you very much for your response. Our question is from YouTube. There are many software available online or as a package for data analysis. What is the advantage of learning a programming language at this point?
So I guess you are referring to the GUI, like the click and point and click softwares. But as I mentioned, it's very nice. Some of the tools, they are very nice, but they're designed specifically for certain tasks, right? Of course, if it can meet your needs, that's fine. But if you learn how to use a programming language, it's much more flexible and much more powerful. You can do any type of analysis that you can, because the software may only has certain functions that you want, and it may take them a very long time to update the software for you to use the other function that you want to use. But if you learn computational biology, you know the programming languages, you can do the analysis anytime. I think that's the advantage.
Thank you for your answer. You can close your presentation, and actually you can see us on also screen if you don't see.
Okay, so let me. Am I still sharing my screen?
Yes, you are
Okay.
No, okay.
I just stopped.
So can you see us?
Yeah, I can see you.
Okay. So I move on with next question. There are now without coding programs coming for future statistics. Is this adequate and permanent to work with?
Oh, I think this is a similar question with the previous one, right? So yes. I would encourage you to actually learn the basics of stats, and sometimes they have like those black boxes, like those softwares you can click, do this stats test, do that stat test. But if you don't really understand it, you may use the wrong test. So I encourage actually you to learn stats, basic concepts, but also if you use a programming language, for example, R, it's really good for stats. So I think still like learning an R package is still R language is very valuable. Yeah.
Thank you for your response. Next question is, how soon can we evolve from a wet lab novel biologist to a computational biologist? Or do we really need to computational biologists for computational analysis?
Oh yeah, that's a good question. Like I was a pure actually wet lab bench scientist. It took me 10 years, but really after the first 1 or 2 years, now you get comfortable with the command line, the R stuff. You can already do quite a lot. But of course, it takes time to be really better, to be good, right? So as the time goes by, you accumulate the experience. I think time is just the currency you have to pay to gain experience. But I would say if you start now, the best time to start is like 10 years ago, right? But the second best time is now. If you start learning it now, I'm sure you'll benefit like sooner, right? So I think if you in my GitHub repository, there are some links for courses that you can take for free. Go there and commit yourself. Every day, do 1 hour, exercise, watch the videos. I'm sure you will get there. So that actually reminds me when I was studying. Yeah.
But wet lab is so time consuming. There is no time to. I'm also a wet lab scientist. But I don't have any time for studying computational things.
Yes, that's also a common actually. So you have to actually better do a time management. So sometimes, for example, if you need to weight something, you're doing the centrifuging, you're doing Wester blot, you can always have those time to learn something. And what I did actually during my PhD is that after work, actually, I would actually watch those Coursera, edX courses at night, and learn the computation and do the exercise. Yeah.
So there is no excuse.
No excuse, yes.
Next question from YouTube. Scalable and reproducible scientific workflows like Nextflow are proliferating day by day. Can we say that computational science is stepping into a new era, or would you recommend sticking to the roots?
Okay, thanks. That's a very good question. Actually, I haven't touched that point. Like workflow language, for example, Nextflow, SnakeMake. I use SnakeMake a lot. Those workflow languages is really useful for pre-processing data. Those steps has like a fixed like step. For example, for RNA sequencing, align the FASTQ. I mean, you do like quality control, then align FASTQ to band files. From the band file, you get this count matrix. Then you go to the D62 for differential expression. For example, those are fixed sets, right? You can easily reproduce, like use this workflow language to actually encapsulate. But really, the downstream analysis require your domain knowledge, your biology knowledge. So that I don't think it can be replaced by any workflow language. You need to, if you're in a PhD program, you know that you always do this analysis. Oh, this is not working. Let me change the analysis. You use a different tool. It happens all the time. So that cannot be automated, and that's why we still have a job, because if that can be automated, I'm out of job. Yeah.
Thank you for your answer. Next question is from YouTube. Data sharing is a big problem. At this point, where can we obtain the data that we analyze to improve ourselves? Does repositories such as GitHub can enough?
Okay, so that two questions here. Actually, the GitHub actually is mainly the repository for code. And if you want to fetch some data, for example, for your own analysis, or maybe even just for practice, go to GEO, and then we'll also have ENA, those like public available. So when you publish paper, you deposit your data there, right? So in my GitHub repository, there are some links there showing, okay, what are the available repositories for data repositories that you can fetch the data. For example, also TCGA, the Cancer Genome Atlas, it includes all the cancer data that you can use for your own project. Yeah, so you go to my GitHub link and you will find some of the links for data repository. Yeah.
Thank you for your answer. Next question. Despite the developments in computational sciences, it is necessary to verify the NGS data with wet lab methods. Should this dependency continue, or are computational methods alone sufficient now? Thanks.
No, I don't think computational methods alone is sufficient. So we always need to do wet lab experiments to verify the finding. But computational biology is very good when you have a large amount of data. You can quickly narrow down the targets that you want to focus on, right? You can quickly generate insights from a large amount of data. Maybe you find the target, then the wet lab scientists need to verify that.
Okay, thank you for your answer. I move on with next question. When I tried to use Docker, it was challenging both in terms of computer hardware and usage. Are there any more user-friendly programs?
Okay, so I think Docker can be clunky sometimes. I understand it, because you have to actually encapsulate all the operating systems. It can be several gigabytes in those, can be really big. So I would say, like, you can use, so nowadays, like, cloud computing is so, it's so easy, like AWS or Google Cloud. You can just increase the amount, disk size, the CPU, the memory. You can always increase. So if your desktop cannot handle Docker, maybe use those cloud computing facilities.
Next question is, if we think of data analysis as a language, would it be more effective to focus on the alphabet or words for learning?
Oh, that's a good question, actually. Yeah. So let me take R for an example because I use R most of the time, right? So you can learn base R, which are the really basic functions, right? I will refer that as the alphabet. But you can also learn tidyverse. It's a group of packages, for data visualization, data analysis, data wrangling. And it's a higher level abstraction of those baseline R functions. So I would say if you want to do something quickly, you want to actually learn the tidyverse first because it can quickly get you some results. But I would say understand the alphabet is also critical, right? Because you want to understand how that high-level function works eventually. And yeah, and there's actually some argument whether you want to learn base R first or tidyverse. But I would say if you really want to get the results fast, you can start with the words. And the other analogy I had is that I want to get from A to point B. I need to drive from A to B, but I don't need to understand how the engine works. I don't need to understand how each of those elements, they all work with each other, right? I just need to know how to drive.
Yeah. Thank you for your answer. Next question is again from YouTube. Tommy, thanks for the talk. Data cleaning part is an important step for analysis. Are you losing any crucial information during cleaning up process? Is there any information showing how to clean data?
Yeah, thanks for this great question. Yeah, data cleaning is an important step, but you are afraid of losing some crucial information, right? So that's why you always need to compare, actually, before cleaning, after cleaning, you need to compare. Is there any leakage, any information lost during this step? And I will say you also need to actually compare the final results, for example, the data without cleaning, the data cleaned, and whether that results make sense or not. So every step, I would say now the domain knowledge actually really actually important because, okay, I clean the data, but now my results doesn't make any sense. Like whether I need to do that or not. So I need to go back to the original step. Okay, maybe let me relax the cleaning criteria. Okay, to see whether the results make sense. So you need to always talk to the expert in, if you're working in biology, talk to them, right?
Thank you for your response. Next question. Nowadays, data analyses are made with machine learning methods, especially for cancer like heterogeneous disease. Mm-hmm. I would like to ask how I need to start to learn them with sufficient R or Python languages.
Okay, thanks. That's a good question. So machine learning is very hot, right? Everyone wants to learn it. So my talk actually didn't touch about machine learning that much, but I want you to start with basic data skills first. And to be honest, most of the biology questions, you do not need deep learning. You may not even need like machine learning. But for your next step, if you want to really learn, there are some areas machine learning is great. And if you want to learn, I would start with, for example, some YouTube channels. Like there's this Josh Stammer has this StatQuest. I'm not sure whether you know this. It's a really good YouTube channel explaining all those machine learning concepts. And there's several other actually courses actually on edX, Coursera that you can take free, and you can just go there and search machine learning in those websites, and you will find courses that you can take for free. So, and I would actually suggest you to use a real data sets. And maybe you can even just read a machine learning paper, like a biological paper, to see how they use machine learning to address that question. And you want to reproduce their finding. And I think that's the best way to learn machine learning and how to use it to address biological questions. Yeah, so get your hands wet. Yeah.
Thank you for your answer. Next question is, if a computational analysis in a published article, which is not reproducible, it is really reliable for the science?
No, if it's not reliable, it's not, yeah, it's not good for science. To be honest, especially if those papers published in those glamour journals like the Nature. I think I saw a paper saying there is a high probability that those papers are wrong in those high-profile journals. So of course you want to see the data, you want to actually see the code, you want to reproduce some of the analysis. Yeah.
Thank you for your answer. Next question from YouTube. What do you think about what should a computer scientist student should do to become a computational biologist?
Yeah. So you're from the other side of the river.
Yes,
okay.
other side.
Yeah. So if you're a computer scientist, so you know programming language really well, but you don't know biology that well. So I would actually suggest you to start learning the basic of biology. There are some courses. You need to understand what's DNA, what's mRNA, what's the protein, how they, and if you want to analyze a type of sequencing data, you need to understand how the sequencing technology works, what this technology is measuring, right? So I would suggest you to understand some of the biology first, and the same advice for people from wet lab. Go find a paper, and download their data. Use this machine learning approach or computational biology approach to reproduce their results, and that will give you some sense how the field how it's working in this field. Yeah.
I think computer scientists have more advantages from biologists.
No, not necessary. I think both have challenges, but I would say learning biology is also not easy, I would say, if you're a computer scientist, because
yeah,
biology is just so weird. Like sometimes it doesn't make any logical sense. It's like why some RNA and protein are not correlated. Why?
Many of us know this.
Yeah, many of you know it, right? So okay, like for
yes.
example, HIF alpha, like the hypoxia-induced effect. mRNA is so high, but why? When there's oxygen, that the protein gets degraded. When you have no oxygen, now it's stabilized. There's so many of those things that biology can explain, but not necessarily the computer scientists that I don't know that. So yeah.
Thank you. Last question. Except from computational sites, what advice would you give to students studying in scientific fields?
Computer. What? So can you repeat the question again?
Except from computational things, what advice would you give to students studying in scientific fields?
I think
not related about your presentation.
Okay.
Just
so
for
I would
academic
say,
career.
Yeah. No matter what you're learning, so it's all about learning, keep learning. So in this case, in this presentation, I show you we need to learn computational biology, learn programming. But if you in other fields, the same thing, you want to keep learning, because the day you stop learning, you start dying, because the knowledge are exploding if you don't keep yourself updated. So my suggestion is keep learning. Yeah, different fields, no matter what kind of fields you are in. Yeah.
Thank you for your advice, and also on behalf of myself and all our participants, I'd like to thank you for spending your valuable time transferring your knowledge to us and for this very informative presentation.
Thank you very much for the invitation, and I hope my presentation is educational and can help some of you to start your journey as a bioinformatician. Bye.
It was really necessary for us. I wish you healthy days.
Sure. Thank you very much.
Bye-bye.
Bye.